Welcome back. As we continue our lesson, we stopped on how to use various laboratory apparatus. You are still with me, George Chapusa, with this biology Form 1. I take you from where I stopped with you. Pay attention. Like what I said in the previous lesson, that biology is worth with the apparatus. We just shared a number of apparatus, and now we are going to look into how we may use these apparatus using their drawings. A biology experiment should be conducted using correct apparatus so that you get the correct results from the experiment. The apparatus should also be handled properly to avoid breaking them. Some are fragile and some require a lot of care for you to use them. Now, let us start with the microscope. How you may use a microscope. A microscope is one of the apparatus which requires a lot of attention as you are supposed to use it in the biology lab. A microscope is an instrument that is used in the laboratory to magnify objects. And a microscope is an instrument that is used in the laboratory to magnify objects. We have already shared in the part 1 which I started with you, but now here I am giving out the meaning of this microscope. The other part of this microscope could be used to observe organisms that are too tiny to be seen using an unaided eye. This microscope can also be used to observe organisms that are too tiny to be seen using an unaided eye. Now, there are two types of these microscopes. There are two types of microscopes. One is the light microscope, and two is the electron microscope. One, the light microscope, and two is the electron microscope. The light microscope uses light to illuminate the objects and the glass lens to magnify the object, while the electron microscope uses electromagnet lens to magnify the object. Of course, these 2 points make the differences with these two microscopes. You may distinguish the two by understanding that the light microscope uses light to illuminate the object and glass lens to magnify the object, while the electron microscope uses electromagnet lens to magnify the object. The most commonly used in schools is the light microscope. The most commonly used in schools is the light microscope. This is commonly used because it is found locally and it is a bit cheaper than the electron microscope. So what we are going to do now, you are going to study. Now I give you a diagram for you to study carefully the parts before I pause you a video to explain about the same microscope. Now this is the diagram of the light microscope, and it is showing you the parts of this light microscope. Now can you study this picture where we have the drawing of a microscope? Thank you for your observation. I think you have thoroughly checked the parts of this microscope. Now, I want you to see this video, which happens to explain more about the parts of this microscope and some of their functions.
This presentation outlines the main parts of a compound light microscope. It also briefly describes the functions of these parts. So, let's begin. The eyepiece lens. It's also known as the ocular lens, and it is the first point of magnification with a 10 times or 15 times magnification. The barrel. It is also known as the body tube. The barrel connects the eyepiece lens to the objective lens, which we'll look at in a minute. The arm. The arm is like the backbone to the microscope. It raises the objective lenses above the stage, and is also the place where we use to carry the microscope from one place to another. The objective lenses. This is the second point of magnification. They revolve around on what is called the nose piece. There are usually 4 objective lenses, color-coded for easy recognition. The 4 times is red, 10 times is yellow, 40 times is blue, and the oil immersion lens 100 times is grey. The stage. This is the platform where the slide is placed for viewing. The slide is attached to the stage by using the slide clips. It is movable in 4 directions by using the stage control. The coarse and fine focus knobs. These move the stage towards or away from the objective lenses, depending on which way it is turned. The coarse focus knob brings the specimen into focus. The fine focus fine-tunes and increases the detail of the specimen. The lamp. This is the light source for the microscope. The lamp brightness can be adjusted using the dimmer. The diaphragm. This is a rotating disc found under the stage. It varies the light intensity that is projected upwards into the slide. The base. This is the bottom of the microscope and is used for support. The base should also be used along with the arm when carrying the microscope. Now, let's review all the parts at once.
Welcome back. I hope you have enjoyed the video lesson where you have seen the parts and their functions. And now I want to present to you a summary of notes from what you have just seen in the video. Now, the microscope, we have the top part, which is the eyepiece. The eyepiece, as the video was explaining, this could be the summary of the function of the eyepiece. So the eyepiece lens magnifies objects. The eyepiece is often untouched and may fall out unless the microscope is kept upright, which means that is supposed to be taken note. You should not be carrying the microscope on the upside down position so that you may prevent the eyepiece from untouched or from falling itself from the microscope. Then it can be removed and replaced with an eyepiece lens of lower or higher magnification. That means that is a piece that can be replaced with any type of a lens that you wish to use at that particular microscope. Microscopes have several interchangeable lenses of different magnifications, such as 5 times 5 or times 10 or times 15. That means those are the sizes of the lenses you may use on the eyepiece. The eyepiece lenses are usually fitted into an opening at the top of the microscope. So this is the demonstration of the eyepiece lens from the diagram or from the video. I think you may now connect with this piece as it is shown to you. Now let us look at the objective lens. This is a magnifying lens. A microscope usually has 3 or 4 objective lens. If you may recall from the video, I hope they happen to show you that microscope had 4 objective lens. So those are the lens you may use as you are looking for tiny objects on the microscope. Their magnifications are generalized as low power, medium power, and high power magnification. Also, the lower power objective lens gives the smallest magnified image. Its magnification is usually written as times 10 or times 15, times 20 or times 25. Lower objective is best used to view the whole microscopic organism. It is also easier to focus and use the microscope at lower power objective lens. Now we have a high power objective lens, which is the best used to view detailed small parts of the organism. So it magnifies an object to times 40 or times 60 to give a larger image. So this is the diagram of the high power objective lens. The high power objective lens. Now let us look at oil immersion object lens. It is a high power objective lens of times 100 or 125, where a drop of oil must be used with the lenses. The drop of oil is placed on the slice and made to touch the objective lens. Now let us look at the stage and the clips. The stage is the flat surface onto which the microscope is placed, the slice is placed, and the stage has a hole so that light can pass through it to the specimen. The body tube. The body tube, the barrel joins the nose piece to the eyepiece lens. It is moved up and down by the coarse and the fine adjustment knobs when focusing the image. Now the coarse adjustment knob. The coarse adjustment knob moves the body tube up and down to bring the specimen into focus. As you want to see the specimen, you use the coarse adjustment knob so that you bring that specimen into focus so that you can easily see when you are using this microscope. So this knob is used with the low power objective. And it is easy to see the tube moving when the knob is turned. Then also on the microscope we have the fine adjustment knob. The fine adjustment knobs, it moves the body tube up and down to the light position so that the specimen is in the sharp focus. It is used to achieve fine focus with the low power objective as well as the high power and oil immersion objectives. The light source. The light source can be an electric bulb beneath the stage, which is controlled by a push button switch. In some microscope, a mirror is used to reflect light from another source, such as sunlight, into the microscope. The light coming into a microscope lights up the specimen so that it can be seen. The iris diaphragm. The iris diaphragm is used to control the amount of light passing through the hole in the stage and through the slice and the specimen. This is done by adjusting the opening and through which light passes. The condenser, which is also called the light director. The condenser is a lens located above the diaphragm, and it concentrates the light before it passes through the specimen. Its position is changed by a knob on microscope in which it is adjustable. Now, that has been a summary of the notes, and now this is the summary of the table. You can start this table as you see the summary of explanation according to what I was just explaining to you. Thank you. So that has been a summary of the notes according to the microscope, the parts of the microscope, and their descriptions together with their functions. Now, we have just learned that a microscope is a machine that we may use in the laboratory, and we have just seen the parts and their functions. But now, assuming you want to carry that microscope to another place, how can you carry that microscope? So the figure below shows the correct way of carrying a microscope. Here there is a person who has carried a microscope, and that is the proper and correct way of carrying a microscope. Now, we have so far discussed the parts, the functions, and the proper way of carrying this microscope. But now, let us look on how we may use this microscope. How we can use this microscope. Now we have the part on how to use a microscope. So, materials that we might need is the same microscope and a newsprint. The procedures that you are supposed to take when you want to use a microscope. One, you are supposed to place the microscope on the bench in front of you. Now, in laboratories, most of the times, you may have benches. And now, place that microscope on the bench in front of you. Then the handle should be towards you. The handle should be towards you. And then make sure that the microscope is not at the edge of the bench. The microscope is not supposed to be at the edge of the bench. Make sure you put that microscope at least at the middle part of that bench. Putting the microscope along the edge of that bench might happen that that microscope can fall down. So look into the eyepiece lens. Look into the eyepiece lens, and you should see. You should observe what you see on those lenses. Now, what do you see on those lenses? From there, adjust the mirror below the stage so that the light coming through the window falls on it and reflects into the microscope stage. Remember, we are looking into the procedures of how you may use the microscope. And from then, cut out a piece of newspaper, print, and place it on the stage. Hold it down with a clips. So what do you see? Or what do you observe? You can read the print on the newspaper. 4. You may read the revolving nosepiece until the low power objective lens clicks into the position. 5. Lower the low power objective lens using the coarse adjustment knob. View all this from the side of the microscope. Number 6, try to look into the eyepiece and keep adjusting the coarse adjustment knob until the print is visible. Very slowly, use the fine adjustment knob to bring the print into sharp focus. Thank you. So that has been the procedure for you to use when you are using a microscope in the laboratory. Next time we meet, we shall look at additional points to note when you are using a microscope. That has been a presentation from George Chapusa. Thank you for being a good listener. Thank you. Until we meet next time. Thank you.